Okay. Good morning. To start this event today, I hope we have a good mood and for this event will run smoothly. I will topic of learning in Bio-informatics from Vidyavardhaka College of Engineering and Mr. Ahmad Zainuri as lecturer from Entrepreneurship Department for opening speech. And I will introduce myself also. Today, and I hope this event will be pitched by Mr. Ahmad Zainuri. Yeah, from Mr. Ahmad Zainuri, the time is
Okay, thank you so much for Miss Novita. Can you hear me, Mr. Novita? Miss Novita, can you hear me? Okay. Okay. Good afternoon, ladies and gentlemen. The Honorable Dr. Joseph Teguh Santoso as a Rector of STEKOM University. The Honorable Professor Pavan Kumar from Vidyavardhaka College of Engineering India. The Honorable Miss Angie Novita Sari as a International Affairs Division of STEKOM University and also all of distinguished guests. Okay, ladies and gentlemen, thank you for each and every one of you being here today. My name is Ahmad Zainuri. I'm lecturer and head of Entrepreneurship Study Program and STEKOM University. Is it such an honor for me to speak on behalf on the organizer? Let's begin by giving you a warm welcome to the event visiting lecture today. Okay, may I first take this occasion to pay tribute to all of the audience of this visiting lecture from Vidyavardhaka College of Engineering India to Science and Computer Technology University or STEKOM University. It is a great pleasure and an honor for me to deliver this opening speech of visiting lecture. May I first take this opportunity to express my gratitude and appreciation as well as extend a cordial welcome to all of the audience in today's event. Okay, next, ladies and gentlemen, today is a moment of extreme pleasure to welcome the guests who are present here at this event of aviational nature on behalf on STEKOM University. In event today, we will learn about every interesting and useful material by all of us from speaker as a professor from Vidyavardhaka College of Engineering India. I hope this visiting lecture today can add the insight and have a benefit for all of the audience as well as provide the approach to solve the problems in the future. Thank you for everyone who have success this visiting lecture event and especially for the student of STEKOM University. Please enjoy this event, take some notes and please ask the question. Okay, in closing, I would like to express my appreciation to the International Affairs Organization of STEKOM and Vidyavardhaka College of Engineering India for making this international relationship. I hope this collaboration can run well and good communication can be established. Well, I don't want to take much of this time again. I will hand over it to Miss Novita as a moderator in this event today. She will introduce our guest speaker to all of you. I would like to say once more on behalf on this seminar organizer. Welcome to everyone. It is a great pleasure to see so many of you here. Thank you very much. Okay, I will hand over it to Miss Novita. Thank you.
Yeah, thank you for Mr. Ahmad for we have Professor Pavan Purman as Assistant Professor of Computer Science. He will presentation about role of machine learning in bioinformatics. Yeah, for Professor Pavan, are you ready for this class today?
Thank you, madam. Here, Mr. Pavan from Vidyavardhaka College of Engineering. I appreciate your cooperation, ma'am. Can I take over the session? Hello.
Yeah.
Can I take over the session, ma'am?
Yes, go ahead.
Yeah.
Yeah, thank you.
Thank you. Can anyone tell me my screen is visible for everyone?
Yeah.
Yeah, my voice is clear. Yeah. Good morning, folks. Myself, Pavan Kumar SP, working as an assistant professor in the Department of Computer Science and Engineering from Vidyavardhaka College, Mysuru, India. Here, I'm planning to discuss few topics that is related to bioinformatics and machine learning. I want to give a small insight towards that combination of bioinformatics and machine learning to all. So anybody have any doubts in the session, you can stop me and you can ask me the questions, or else you can wait for at the end of the session, and that time you can ask the questions whatever you have in your mind. I hope this is clear. So my discussion topics are like this. Yeah. So I'm going to have discussions on first, let's understand what is bioinformatics. Then we'll take up a few topics on machine learning, like what do you mean by machine learning, where we can use the machine learning, and what are the branches of machine learning. That and all we'll discuss. Then we'll take a few applications of machine learning in the field of bioinformatics. And then we'll talk about applications of machine learning in healthcare. Then we'll carry out the same discussion with few case studies. One is what protein structure prediction, and one more is what protein and aptamers protein interaction, prediction of aptamers protein interactions. I hope my discussion agenda is clear for everyone. Let's begin with the bioinformatics. First, we need to understand what do you mean by bioinformatics. In the name only, we have a hint. Like we are talking about biology, means that concept which are related to a biology, and we have informatics over there. Means what we are really trying to do is we have a biological data. It may be a sequence. I think everybody know the RNA and DNA sequence, right? RNA, DNA sequence, or a genome sequence, or you may have an image, or else you may have a text which is related to a biology, or else a patient records, etc. We can call it as biological data. So, and we have few algorithms. We are going to use that algorithms to compute something which is related to this biological data, or else we can say like this. We are going to apply the computational techniques or a computational algorithm on this biological data, what we have here. Or we can also apply the analysis technique or analysis algorithm on this biological data. When you have a combination of computation and analytical algorithms or computational analytical techniques we are going to apply on this biological data, definitely we'll end up with few inferences over here, or we can end with one interpretation. So this is what bioinformatics. So here we are involving the biology, and we are involving the computation algorithms, and we are involving analytical algorithms. So put together, we can call this concept, or put together, we can call this domain as bioinformatics. I hope everyone is clear with the definition. So everybody know this bioinformatics, biotechnology, machine learning. Everybody is having the idea. Just I'm briefing a small thing which is related to bioinformatics. Then this bioinformatics, what we have here, it is a completely interdisciplinary field. Means it is not restricted for only for biology. Look here, you're going to include computer science over there, you're going to include biology over there. Here we have a statistics, and we are working on genetics, and we are working on mathematics. Okay, so we have 4 to 5 fields. We can call it as bioinformatics. It's completely an interdisciplinary domain. So anyone can pitch in and they can contribute in the field of bioinformatics. It is not like that, okay, only the engineers, those are doing the PE, BTechs, graduation engineering, they can do the bioinformatics. It's not like that. So basically, I'm a computer science engineer. So, but I works, my research topic is like bioinformatics, protein production, and science. It's completely interdisciplinary project. And everybody knows that without mathematics, you cannot perform machine learning. Each and everything is in the machine learning is completely depends on the mathematics and mathematical formula. So definitely you have a small entrance and the basics of mathematics you should know before entering into this field. So together we can call this as an interdisciplinary subject. So everyone is have an equal chance to contribute towards research in bioinformatics or biosoftware. So then we'll continue our discussion towards machine learning. It's a favorite area and it is a fancy word also. So everybody wants to do something in machine learning. Before going to do something in machine learning, we need to understand what it is exactly. So here we are teaching a machine, or else we are making a machines to think by itself, or is taking a decision on its own. And we are not programming it again and again. Once it is programmed, it is done. So all the experience issue what it can have in its daily life, it will learn from that experience, and it will be ready for today and tomorrow. That is what the mission. So everybody knows it. For example, when we were kids, our mother and father used to feed us like at dinner time or at lunchtime. Then when they are feeding us, always they will show something to us. For example, some dog is roaming over there. So the mother will show always a small kid, okay, look, that is a dog. One day, the mother told the same thing, yes, this is a dog. The next day, yes, she showed the dog and trained her baby like, okay, this is a dog. On the third day, baby itself will say, okay, this is a dog. Look out, what is happening here is the baby is training by looking at the visuals like the dog should have four legs, it is having a tail, and it makes a noise like bobo. So that and all it analyze and it will have its own hypothesis in its brain. Like something is coming in front of it, which is having a four legs and a tail is firing like this, and it says Bobo, so it is a dog, a small kid. The same way, what we're trying to do in your machine learning is we are trying to train our machines by looking at the features. By looking at the features, it may be a camera inputs, or it may be a sound, a noise, or its action, so anything. So by looking at the features, by looking at the environmental status, we are going to learn something, or means we are training the system to learn something. That is what we call it as machine learning. So let's continue our discussion about this machine learning. Here we have few branches of machine learning. One is supervised learning, another one is unsupervised learning. Third one is what? Reinforcement learning. So let's talk about the supervised learning. So when we are now I already discussed about what is machine learning, right? So now I'll talk about supervised learning. So what do you mean by the supervised learning? Okay, when we are training the system, or else when the system is getting trained, some supervisor should be there. Like when you're solving, for example, so you're solving few problems in mathematics. So you have solved the first problem and you check the answer for it. And you have solved the second problem and there also you check the answer for it. You solved the third problem, you have check the answer for it. At the 11th problem, you don't have the answer, but the answer is correct. Why? Because by solving all these examples, what you have here, by looking at the answers, what is an expected answer? Everything you know earlier, what you're doing, you're creating your hypothesis, or else you're creating your concepts. At the end, the 11th problem, if you don't have the answer also, you can predict the correct answers. Like examination preparation, before the examination preparation, what you'll do actually. So you guys will keep on solving some mathematical problems and you'll prosperify your answers. So until unless you are getting a correct answer, you keep on repeating the process. That process we call it as training. When you enter the examination hall, or else when you're writing the examination, in that case, only the question papers you have in your in front of you, but there is no answer. So the concept what you have learned during the practice, the hypothesis what you have learned during the practice, that makes you to solve that problem in your examination. If you got 100 out of 100, means you have learned it properly. If you're not getting 100 out of 100, you're not learned properly. So you need to go back and you need to train yourself. So some supervision will do it. Means we know the expected answers. We have the instances, we know the expected answer, and by having these two things, we are trying to train our networks, or we are trying to train our model. So that is what we call it as supervised learning. So now what are the algorithms? We have the neural network, we have the decision tree that can do the supervised learning. We know the answer before the tree. The same way we have unsupervised learning where we don't have any supervisions. We don't have any supervisors. So like testing algorithms and all, it is unsupervised learning. So nobody is going to supervise. Exactly like your life. So good and bad things, it's learned by you. Nobody told you like this is good and this is bad. And with your life experience, only you come to know that okay, what is good and what is bad. No one is supervision. Okay? Nobody is supervising. So this is what we call it as unsupervised learning. Look, both the supervised and unsupervised learning will help us to solve some complex problem in bioinformatics. So then we'll talk about the reinforcement training. The reinforcement training is a trendy version of machine learning here. So in reinforcement training, what we have an agent. We'll be having an agent, and the agent will learn something from the environment, and it is having a goal state. So for each time, by every time it will learn something, or every time it will take some input from the environment, or it sends some input from the environment, it makes its own decision. That decisions or that actions carries this agent to the next step. So every positive moves, every good move, the trainer will reward a positive point to it. For every a bad move, it will assign a penalty for it. Exactly when you are in schools. So if you got out of marks, my for means 24 25 marks, you got 25 marks, your parents are very happy, they will treat you. If you're getting a less marks, they're not that much happy. So that is what negative reward. Think that you guys are agent, your classmates are envy. So in your environment, means in your classroom, whatever inputs you can get, look, your staff or instructor, they will instruct some concepts to you. So that is what the input you are taking from the environment. So you have a senses. What are the senses you have? You have eyes to take a visual inputs. You have ears to take an audio inputs. Correct? No? So you guys are like agent in the environment class, and you are taking the input from the environment. At the end, you are going to write your examination. If you are getting out of marks, means 30 out of 30, 100 out of 100. So definitely you will be rewarded by a positive point from your parents. Otherwise, you are going to get a negative reward. So what this negative reward will affect? Yes, the negative reward will affect you. Why? Because always you have to be very happy. Always you want to keep your parents happy. So the negative reward will push you to perform better. Getting your point? That type of concept we call it as reinforcement learning. So these are three main branches we use in your bioinformatics. In next slides, I'll discuss briefly about that. And here, let's take up one by one. And now I'll talk about the genomes. So it is a sub area of your bioinformatics where we are essentially using the machine learning algorithms to enforce something. So first thing is what we need to understand what is this genomics. A genome is a complete set of genetic material found in atoms. Look at it, it forms in your chromosome, the longest DNA. It is somewhere it is there. So what it will tell us that each and every functionality of an organism is depends upon what this gene, what is the behavior. The genes we normally we can call it as genes. So now you can ask me, sir, we are computer science students, but you are talking about the biology. So how you can correlate it? Because the genes and genomes, we have a sequence. By using the English letters only, we are writing the sequence of this gene, human genome, any organisms, it have its own gene sequence. By analyzing that one, we can infer something about it. And many diseases can be identified. Genetic disease can be identified by the analysis of this genome sequence. So here we request the computational power. Here we request the matching algorithms. So when the process it takes much time when you're doing with the what we can say a laboratory process using some chemicals or something. Like normally in the field of bioinformatics or biotechnology, it costs more. Like when you're predicting a proteins only by using extracrystallography method, so it takes much more dollars to complete it. So by using this machine learning detection, we can reduce the cost of those kind of problems. So here now I'm talking about the genomics, right? Yeah. So here this genomics, we are talking about the genes, and we'll be having the sequence over there. If we have a sequence, we can view this problem as an NLP problem or a text mining problem. Yes. So the genome is a study of the genome mapping, evolution, and editing. Like here we are studying about the genome mapping, like how it is getting mapped, how it is getting evaluated, how it is being edited from years to year. So here in the genomics, we have three subsets. One is what regulatory genomics, and second one is structural genomics, and third one is what functional genomics. What it is exactly? First, I'll talk about the regulatory genomics. So it only concerned about how to regulate the genome expressions. So in machine learning, how you can, in the field of machine learning, how you can improvise it. So one thing is what you can produce RNA binding proteins. So the binding will happen. Okay, when you have two proteins, when you have a codons over there, you can easily tell like which one can bond with which one. So what we are trying to do in your machine learning is here what I'm writing is completely where we can use the machine learning. Okay, the first one is producing the RNA binding protons. Then second thing is what transcription factors. So you can predict the transcription factors also, and then you can predict the classifying the gene expression. Look here only what we are doing. Look here we are doing the two things. One is what predicting, another one is what classifying. The machine learning we are doing the same thing. Right? We are performing the classification. We are performing the regression. So all the concept what we have learned in the machine learning, we can use in the genomics, in the regulatory genomics. So we can predict something, we can classify some genomic expressions and all. So it is possible, and lot many papers are got published on this source. You can search in your IEEE, ACM, so you'll get hundred of papers where the machine learning is applied on this genomics. And it is a very good area. You can publish a very good papers over there, and it's completely interdisciplinary. But you should have a minimum knowledge with respect to your biology and gene genomics and all. And the second one is what structural genomics. Structural genomics are concerned about only the genomic structure, how it is getting structured. Structure means how the sequence are getting organized. That's what we call it as structure here. We're talking about the structure, we are studying about the structure, we call it as what? Structure. So here, how we can help this? So here we are trying to classify or predict the protein structures. Everybody know what is protein and the structure of protein. Everybody know. Okay, that is what my case study in your next slide, I think. So I'll discuss that protein structure prediction. A very wonderful concept, and the deep minds, that community, they have done a great job with respect to this protein structure prediction. So I'll show you the website and all. So you'll get an insight of it. So then we'll talk about the functional genomics. In functional genomics, the functional interactions. Because when you have a gene, when you have a small biological complex molecules like DNA, RNA, protein, enzymes, so they will interact with each other. They will interact with each other. When the bacteria is covered by a surface protein, for example, you can take the example of your COVID. Okay, always when the COVID-19 enters the body, there is a receptor in your body that will catch that protein. Not simply it will catch that, but because there is a bonding is happening between the receptor layer and the surface protein of the coronavirus. If they are ready to bond, yes, it will take the coronavirus inside. That's how it works actually. So there is an interaction will happens always in between the biological complex molecules like DNA, RNA molecules, or as proteins, enzymes. That is a use of that one, right? So okay, so where you can help us, yeah. So here we are trying to classify the mutations or protein structural or localization. So many more things is not possible in the field of biology, or else the problem statement which are taking much time by following the biological process that can be easily done, that can be easily solved with the help of your machine learning process. Normally, as a human being, yes, we can think of a new solution, but when you have too much complex problems like n dimension problems, 10-dimension problems, you cannot do it manually. So we have to go with the machine learning problem, and the deep learning and the neural networks, they are contributing more towards this genomics. So we'll take up that one and discuss. Okay, I think somebody raised the hand. We'll start with them. So can you unmute yourself and ask me what is your doubt? Rajesh, Worry. Yeah. You can unmute yourself and you can talk to me. Rajeshwari.
Hello, sir.
Yeah, Rajeshwari.
Okay, sorry for interrupting, but
yeah,
it is
no issues.
Yeah, it is your voice is keep fluctuating, sir. So it is bit noise actually. I can't hear clearly. So it makes me quite difficult to understand.
Okay, from so from past 5 minutes or past 10 to 20 minutes, no?
I think it is since the first.
since from the starting, huh?
Yeah.
Oh, sorry,
But
sorry.
it's fine. It's fine. It's fine. But yeah, can you make that noise reduce or something else?
I'll rejoin the session. Huh? Give me 2 or 3 minutes. I'll rejoin the session. I hope you're getting my points, Ishwari.
I quite understand. Yeah.
Yeah, if anybody have any doubts, you can stop me and you can ask me the questions. I'll rejoin the session. Okay, give me 2
Okay.
minutes.
Okay. Thank you, sir.
Yeah, thank you, Mahesh. Can anyone tell me my voice is clear now? Can anyone tell me my voice is clear?
Yes.
My voice is clear.
Yeah, it's clear.
Now it is clear. Shall I continue?
Clear, sir. Clear.
Yeah.
Yeah, absolutely.
Thank you for interrupting me.
Mhm.
Okay, you want me to repeat the concepts, guys?
Can you repeat the genomics?
From what slide? Slide number.
Genomics part from regulatory until functional genomics.
Yeah, I'll go back to the previous slide and I'll continue. Thank you.
Okay, thank you, sir.
Okay, yeah, we'll work with this genomics. I have told you, right? Like every expression or else your behavior, your parental behaviors, everything is depends upon the genes what you are carrying, right? So the information related to your organism, the functionality which is related to your organisms, everything is related to your genomics only. So here we have some few subsets of genomics. One is regulatory genomics, another one is structural genomics, third one is what? Functional genomics. Functional genomics. So let's explore one by one. Yeah, first one is what? Regulatory genomics. Here we are only concerning about how to regulate the genome expressions. The regulation, you know, right? So there's strictly some rules over there. How you can regulate the genomics, how you can regulate the genome expression. Then here, where you can apply the machine learning here. So if you want to produce RNA binding proteins, means you can use this MI techniques or machine learning techniques to do that one. And second thing is what? Transcription factors. If you want to predict or classify the transcription factors, yes, of course you can use the machine learning techniques over there. The third one is what? If you want to predict or classifying the gene expression, yes, in that case also you can go with what? This machine learning techniques. Then you have a structural genomics. So here in the structural genomics, we are concentrating about the structure of genomes, like how the sequences are aligned. So we are concerned about that one. So in that domain, how you can help the scientists, how you can help the community with the help of your machine learning is, yes, you can apply some machine learning techniques to predict or classify a structure of genes. Yeah. Okay. In the same way, we have a functional genomics. So here we are talking about the functions and interactions of your genomes, like how it is interacting. When you have a protein-protein interaction will happen, the protein and enzymes interaction will happen. So everything will happen with respect to this. So we are concentrating on the function and interaction of these genomes. So in this domain, how you can help or else how you can contribute is, you can apply some machine learning algorithms to classify the mutation and protein cellular localization. So we can apply all those things here. Students, give me a second. Okay, sorry guys, there was a technical glitch. Yeah, fine. I hope you understood this way you can apply this machine learning algorithm in the field of genomics. Then take up your case studies. Yeah, before going to that one, let's talk about few more things about the application of machine learning in bioinformatics. So one is what? Proteomics. Okay, can anyone tell me what is this? You can raise your hand or else you can talk to me directly. So anything is fine. Kindly unmute yourself and I need an interaction from your side so that we can discuss few points properly. So can anyone tell me what is this diagram? Let me see the chat box. I think I'm not getting anything from you guys in the chat box. Can anyone tell me what is this? Yeah, it is a folded DNA. No, it is not a folded DNA. It is a tertiary structure or secondary structure of your proteins. The proteins what you have in your body, it is a structure of it. Can you see this? This is structure of protein. What you are seeing in the screen. Yeah, yes, Rajeshwari. It is not a DNA. It is a structure of your protein. So here in this field, what we are trying to do is we are trying to study the proteins. So what do you mean by the study of proteins? The proteins means what are the components of your protein, how the proteins are interacting with each other, and what are the roles of that protein in your body. Like in your body, if the new cell is generating or else something is growing in your body, for each everything you are respiring, for each and everything the proteins are responsible. So you have lot many proteins, and each and every protein it has its own different structures. So that is completely comes under your protein structure predictions topic. So that is what my case study. I will show you how the protein looks like in a primary structure, secondary structure, and in your tertiary structures. So we are applying a machine learning technique in this proteomics also. So we are working on the proteins, protein structures, protein-protein interactions that and all we'll take and we'll discuss. Okay. And in your healthcare, where you can use this machine learning. Because the machine learning and artificial intelligence are being used extensively in the healthcare facilities to improve the patient case and enhance the quality of life. Okay, I'm not telling about someone should be ill, then we need to treat them with the help of this machine. No. Okay, how you're going to treat. So, for example, somebody is having some deficiency, you need to apply this machine learning technique over there. You need to come over, like, what are the deficiency over there? You need to understand that. Then you can send the supplements over there. Or else somebody is having some genetic disease, you need to identify that. Some proteins are not working for well, or else look here. The functionality of protein is always depends upon the structure of the protein. If it is not structured well, you cannot expect a functionality from protein. Yeah. So that and all we discuss. Then some of the main application of machine learning algorithm in healthcare includes drug discovery and manufacturing. So we are using this machine learning to identify the drugs, or else manufacture of the drug discovery. We are using this machine learning algorithms, maybe supervised or unsupervised learning algorithm for a drug discovery. Then medical image and diagnosis. Look here, this is a vast area. Look here, we have many kind of inputs like CT images, MRI images, so medical imaging or else microscopic images. Like lot many papers have got published to identify your lymphoblast or lymphocyte. The input for it is what that microscopic images. So they look into that cell and the cell shape and the diameter of the cell, the irregular shape of that cell. They'll identify what is lymphocyte and lymphoblast. So here, what is the input actually? The input is what the image. So it may be a CT image, or it may be a MRI image, or else microscopic image. We are going to have that one as an input. We are trying to apply a machine learning algorithm to it, and we'll get some inference out of it. So a lot many papers are published on your COVID-19 also. So many of the papers got published like identification of identification of COVID-19 by looking at that CT images, by looking at the MRI images. Not many papers have got published. You can have a look in reputed journals. You'll get a papers over there. What is the input actually for us? For us, the input is what? Imaging. Yeah, that is what my second thing. So where you can apply the machine learning techniques and personalized medicine. Look here. Normally, if I got a fever, if you got a fever, both are treated with same Dolo 650, right? Same paracetamol. So it is not a personalized medicine. It is a general medicine. So anybody got fever, we have to treat them with a paracetamol or something, correct? But what do you mean by personalized medicine? Look here, in your medical field, not all the devices are general. So very rare diseases are there. Very rare diseases are there. For them, we need to treat the person personally. We need to understand. We need to understand the responsiveness of a body, or else we can say like patient friendly or patient personalized medicine. We need to create it, or else we need to manufacture it. Then only we can treat them. So that's what we call it as personalized medicine. If you want to perform that, if you want to manufacture that personalized medicine, definitely you have to go with the machine learning techniques, and a lot many machine learning techniques are applied on this area. So then the stroke diagnosis also, we can take a help of your machine learning algorithms, and definitely we will take a biological data for machine learning model. We'll try to classify, or else we'll try to predict some of the inferences. Okay. Now we'll talk about the case study. What I have chosen here is first one. I'll take up pure protein structure prediction. Second thing I want to talk about aptamers. I want to talk about aptamers. I don't know how many of you know the protein structure, DNA, RNA, and aptamers. So I want to ask you, how many of you know the basic knowledge about the protein and protein structures? In the chat box, you can answer, or else you can unmute yourself and you can answer. Anything is fine. Students, I'm asking you.
Yeah.
Yes. Okay, in the chat box, I have few answers. Basic BSc, I was a science student. Very good. In my senior high school, so yeah, very good. So most of you know what is protein and all, right? Any protein example you can take. Here, before going to a machine learning topic which is related to protein structure, first you need to understand what is protein and what is protein structure. Look here, the protein is always made up of a sequence of amino acids. Totally, we have 20 to 21 amino acid sequence. So all amino acid sequence are put together, it make a chain. This we call it as primary structure of a protein. You can see here. So we have a protein, we have amino acid sequence here. You can see A, R, N, D, C, Q, E, G, H. So this is what my amino acid sequence. They are put together, they are sequencing. At the later stage, it will be converted as what? A secondary and tertiary structure of your proteins. I hope this much is clear. So you can see the amino acids here. Yeah, let's go one by one and discuss what it is. Look here, the proteins are nothing but a large biomolecules composed of one or more chain of amino acids. Look here, it is a large and complex. We need to mention that one also. It is a large and complex biomolecules. It is composed of one or more chain of amino acids. So we have a chain of amino acids. It may be a single chain of amino acid, or else it may be a multiple chain of amino acids. So the second point you need to remember is each protein is made up of different structural combination of 20 amino acids. Totally put together, we have 20 amino acids. We are going to rearrange the amino acid one after the another. That becomes what? A new chain of amino acid that results a new protein for you. Then each of the 20 amino acids has a specific side chain known as R group and also attached with alpha carbon. So this group we call it as what? R group. This group, each and every amino acid. Look here, this is nothing but a single amino acid. It may be any amino acid. Look here, this amino acid is what this. So it is structure of a single amino acid, the general structure of a single amino acid, we can call it as. So it will be having a R group, and it is also attached with what? Alpha carbon group also. So this is quite common in all amino acids. And R group has a variety of shapes and size changes the responsible of biochemical reaction. Look, everything is almost remains same, but we have a variation here that makes a change and a change in the shape and change in the shape of your protein. Means when the amino acid sequence is converted into a secondary structure, it will be having two structure like alpha helix and beta sheets and all. So it will turn, it will twist. Why it is getting twist? Why? Because the amino acid will interact with other amino acids. So when the interaction is happening, there is a chances of getting pi bond, psi bond. So few more bonding will happen. That cause a new structure. That cause amino acids are getting twisted and it is converted into a secondary structured protein. I'll show you. Then the chemical composition of a side chain determine the characteristics of amino acid. The side chain what we have here, that will determines the characteristics of amino acid. You can ask me, sir, what is the characteristics of amino acids? Like the few amino acids, they're hydrophobic. Few of the amino acids are they are hydrophilic. So these properties of amino acid are the main feature to understand how the proteins are getting folded. Again, in a moment, we have 15 to 20 properties of each and amino acids. That will be the feature for us. In your machine learning, or else we'll talk about the supervised machine learning over here. Look here, if something is happening, so we need to analyze the features. Means we are going to analyze or we are going to create a hypothesis by depending upon the feature and what we are trying to do. We are inferring something, or else we are predicting the output over there. Here also, the side chain of the amino acids that is responsible for the characteristics of amino acids. Some of the amino acids are hydrophobic, some of the amino acids are hydrophilic. So these properties will help us to predict the structure of protein. So not only these two things, we have many more properties. We need to go with charge and all. So we need to work on the energies. So we have more and more properties over there. We are going to use all the properties. We'll try to predict the next structure of this protein. Till now, we are talking about the primary structure of protein. Then after folding few things, few amino acids, after binding few amino acids, we'll get a secondary structure. Then we'll get a tertiary structure. Then we can get a quaternary structure. That is the final structure of your protein. Yeah. Now these are the properties we have here. Okay, see here. Can you see this? This is a sequence of your amino acid. And we can also call it as primary structure. So this is what your one amino acid, and this is what your one more amino acid. This is what your next amino acid. Like this, you have a chain of amino acid. This we can consider as the primary structure of your protein, sequence of amino acid. Right. This next, each amino acid are interacting, means they are creating some hydrogen bond, they are creating some ionic bonds. The same chain is twisted like this. So this formation we can call it as what? Your secondary structure of protein. Then again it is getting folded. So that will be considered as what? Your tertiary structure of your protein. Then you have a native structure of your protein. When, look here, there is a journey from the primary structure to a native structure of a protein. Look here, during this journey, we have lot many folds are happening, lot many bonds are happening. So many more things are happening, complex chemical processes are happening from your primary structure to your native structure. When we have the native structure here, so this protein is completely functional. You can call this protein is completely functional. If something is not wrong, it is not properly structured, the protein what we have synthesized, it is useless. It will not function at all. And here we have a mystery. Okay, if you have a same sequence of amino acid, when you put together, always you will end up with what structure? The same structure. But we don't know how it is. It is a mystery over here. So how we have solved this mystery? So there is a community called AlphaFold. They have solved this mystery. Let me show you that website. What they and let me show you that the website. Yeah. If you want, I'll put it in your chat box. Look here, this AlphaFold protein structure. Look here. Very recently, they have achieved almost 96 to 98 accuracy, I think. They have predicted exact structure of your protein. Actually, 10 years back, it is was completely impossible to predict the protein structure. But these group, the DeepMind group, they have done it. So you can go through this website. You'll get complete information what they have done. Look here, AlphaFold is an AI system developed by DeepMind that predicts a protein 3D structure from an amino acid sequence. It is regularly achieves accuracy compared with the experiment. Look here, they have done it. Actually. Look here, it's a very tough job when only you have amino acid sequence and you are expecting a 3D structure, means the final structure, native structure. So it looks like impossible, but they have done it with the help of machine learning. So they have released a paper also. You can go through it if you are interested in it. There is a paper, you can go through it, and you can check the methodology also, how they have done it. Because this is a paper, you can go through it, and I'll show you only the methodology what they have used. Look here. Can you see this? These are the experiments what they have conducted. And this is the architecture of their neural network or deep learning. Can you please go through this paper? I try to understand how they have done this process. So this is what the architecture. You can see the architecture, what features they have taken. how they have processed each and every nodes and all. So complete details explanations are given. In this chapter, you can see. Okay, let's come back to our discussion. Anybody wants to discuss anything? So I'm here to discuss. So then I'll take up my next case study, the prediction of aptamers protein interaction. So you want me to continue? I will go through it. Otherwise, I will answer your questions. If anybody have any questions, they can ask me. If no questions, at least put it in the chat box that you guys are not having any questions, so I can move forward. I will ask later. Yeah. Very good. Shall I continue now? Thank you so much. Okay, now we'll talk about the aptamers. So what do you mean by aptamers? Most of you not heard about this aptamers. It is nothing but, look here, the aptamers are a short nucleic acid sequence compatible with specific high affinity model binding. It is nothing but a single stranded DNA. So it is nothing but a single stranded DNA as very simple nucleic acid sequence here. Why we are using the aptamers? Yes, we are using the sequence of your DNA, single stranded DNA, RNA, we are using it here. So why we are using it? We have few applications on it. So what are the applications? Look here, we are using this aptamers to diagnose some diseases. We are using this aptamers to inspect some food, and we are using this aptamers for the drug discovery and therapeutic tools we can use as a therapeutical tools. So what do you mean by aptamers? You can see in this diagram what I have given here. It is taken from this website. So this is what a protein already. Look here, this diagram what we have here, it is a protein. We can call it as a target protein. And we are trying to bind the sequence what we have chosen around this target protein. So the aptamers always will use to bind the target protein, or a target protein. It may be on a bacteria or it's a microorganisms. To identify that one, we can use it. So today I'll take this food inspection topic as what my case study. I will show you how you can go with the aptamers and how you can design your aptamers. I hope this is clear. So here we have a target protein and we have an aptamer here. This aptamer is nothing but what a single stranded DNA or else a simple sequence over there. This aptamers, we are going to choose an aptamer in such a way that it should bind very properly around your target protein. Yeah. Now, normally in chemical biological process, how they are selecting an aptamers? That is the question. So what they will do actually, now everybody know the E. coli bacteria, right? So that will cause cholera for you. E. coli bacteria that is responsible for cholera. So what we do normally in biological laboratory, we'll take the sample of the E. coli bacteria. Lot many samples we'll take. So once the samples are taken, what we'll do, we'll feed this bacterial sample to the aptamer pools. Aptamer pools are nothing but a different combination of your RNA and DNA sequence. There is no names for it, like millions of sequence will be there inside this aptamer pools. So what happens, always the bacteria, the surface protein is there. So this aptamer pools, one or the other sequence will attach with that bacteria. So for example, when you have a sequence like this, you will be having a bacteria. The bacteria will have what? Surface protein. So this RNA DNA strands will bind on that surface protein. Bind on the surface protein. Not all. So few of. So it is nothing but what we are trying to do is, here we have a lock. We lost the key. So here we have set of keys here. We have some set of keys here. So what we are trying to do is, we are trying all the keys what we have here. It is nothing but the process. So we have a biological bacterial sample and we have an aptamer pools here. So we will put all the bacterial samples to the aptamer pools. One or the other aptamer will bind with that bacteria. Means when you have a bacteria, what it will do, the sequence will bind properly to it. Then we will wash it. Look here, this is a protein, and this is what the aptamer sequence, what is binded very properly to the protein. So after binding, we'll repeat the process. We want the confirmation, right? Whether the aptamer sequence is binding very properly or not. So we keep on repeating this process again and again, and we'll wash it. And once we confirm that, okay, this is a sequence of aptamer, it is binding very properly, we'll make the PCR, means we are making the multiple copies of it. Once it is done, Once the PCR is done, so then what we'll do, we'll add a fluorescent into that sequence. We'll add a fluorescent into that sequence. Then this what we have created the sequence. Now we'll put it on the food. So here it is your food. We call it as burger, right? So we'll put the sequence on this burger. If we have a E. coli bacteria in this food, so it will glow actually. Why? Because we have added a fluorescent to it. So this is how we use the aptomers actually in biological process. So the process what I have shown you here, we call it as Select process. We call it as systematic evolution of lesions of experimental enrichment cycles. So simple. Here we need to identify a proper sequence that binds on a target protein. That's what we are doing. That sequence we call it as aptomers, and very properly binding sequence we call it as what? Aptomer here. So here this is an example how you can use the aptomer to find out the traces of E. coli bacteria. So now the same process will cost much money. So what we can do, instead of doing all this process, just study the surface protein structure of a bacteria. Surface protein structure. This is what the bacteria, right? It looks like this. So it will be having a surface protein. Try to understand the structure of it, and you need to predict a sequence that should bind on this surface protein exactly. If you are capable of doing that one, this much process need not to do, and you can reduce the cost also. So in this phase, you can use a machine learning technique to identify the aptomer sequence, or else we can say predict the aptomer sequence that binds a surface protein. So here we can use a machine learning algorithms. So I think very few works has been done on this process. If anybody interested, you can take up this work and you can do it. So what are my observations when you go with this biological process or a biochemical process? So it requires highly skilled expert. If you want to perform this, it requires highly skilled recruits, and huge processing cost is there. And there might be a cross reaction of aptomers. Why? Because when you have too many aptomers in a single pool, there is a chances of getting cross reaction over there. Yes. Then we have a complex procedure to get a specific aptomers. These are the drawbacks what we have right now on your biochemical process. So everything can be overcome with the help of your machine learning that we need to think and we need to do. So at the end, you have few questions like, okay, so where we can get the data? So on what data we need to work, correct? So here I have mentioned few databases over here. There you can get the genome sequence, you can get RNA and DNA sequence of some bacteria. COVID-19 also you can get it. So any proteins you have, you can go with the structure, you can go with the sequence of it. Protein sequence you can get. Here I have mentioned few databases here: European Molecular Biology Laboratory, EMBL, we call it as National Center of Biotechnology and Information. We not standardly we can call it as NCBI. So in NCBI you'll get all the sequences in FASTA blast code. You can download it and you can do whatever you want with it. So you have PubMed, there is a link I have mentioned here. You have a gene bank there, so you can go to the gene bank and you get the sequence over there. Then you have a DNA data bank, you have genome information broker, we call it as GBI. Then gene link you have, molecular probe you have. So these are the database already existed. So you can go to the database, you can get the sequence and apply your machine learning techniques and try to infer something about it. So these are the sources I have referred for this class, or I referred for this discussion section. So finally, I want you to show, look here, till now I have shown you theoretically, like always I use this word sequence many a time, right? So how the sequence looks and how you can process it, how you can convert the sequence into a structure. That and I can show you practically now within next few minutes. I'll stop sharing because I want to navigate to another screen. Is there any example of aptomer processing, sir? Can I get it fully? Yes, I can give you. Now I will show you how you can get a data from one of the website, one of the database. Everybody know the Google Colab platform? Anybody know the Google Colab platform? Yeah, you can text it here, I think. Okay, fine. Let me show you one thing I have done with respect to the COVID-19 virus. I can show you that. Give me a second. Yeah, very good. That is a good platform to code. Okay. Fine. Python programming language. Yes, it is for Python programming language. I can show you something. Yeah. I think you can see the screen. Can anyone tell me about the screen?
No. Okay.
Yeah, I think. Give me a second, students.
Internetnya ih.
Sure. Hello.
Yeah, it is visible. Thank you. Thank you.
Yeah. May I know, is it enough?
What happened?
May I know, is it enough for presentation, or maybe you have some slide?
No, I don't have any slides. I'm showing it like how you can use a BioPython module.
Okay, you can continue this. Thank you.
No, I'm still continuing. Practically, I want to show the students like how you can use BioPython here. Here, I'm using a BioPython module. You can install it by using this command, install BioPython, or else here I have mentioned few commands. So lot many videos are there in YouTube, or else it is completely available how you can analyze COVID-19 data set. You can refer any materials, books are available or videos are available in YouTube. You can refer and you can learn. So here I'm trying to install that BioPython module. You can see it is about installed in my system.
Yeah.
Once it is installed, you can check what are the methods are available over there. Then we are trying to get a sequence here. Before going to that COVID-19 sequence, let me show you how the sequence looks like. Look here, what I have written here: ATT, AGC, ATGC, GTA. This is what the sequence. What kind of sequence it is? It is a DNA sequence. What I'm showing you, it is a DNA sequence. I'm not telling what DNA sequence here. Yeah, sample DNA sequence. What I have here. So you can take up that sequence, you can store in a variable, and when you have the sequence, okay, if I ask you to write a code, find out how many A's you have, how many T you have. So this ATGC, what I have written here, it is a code answer actually. I'll show you at the end. So what do you mean by that? So look at this DNA sequence I have extracted from some textbook or some material. So I'm storing in a variable my DNA. So if you want to count how many A's you have in that. So now it is not looks like a problem which is related to a bioinformatics. It looks like we are working on NLP. We are working on a text mining, right? So we have a sequence over here. So in that sequence, if you want to count A, yes, here we have a direct function, direct method to do it. Okay, we have a direct method to do it. Yeah. So you have a few more methods that helps you to analyze a sequence in your DNA. It may be a DNA sequence or a RNA sequence or protein sequence in your BioPython module. In your BioPython module, you can see here. So directly I will go to that COVID-19 data set. So what I have given you that ATTGC, right? So this you can refer here. You can call it as codon table. You can call it as codon table. So each and every amino acids what we have, it is have its own code. Each and every amino acids what we have here, we have its own code. So when the protein is getting synthesized, there is a sequence of steps should be performed, like transcription, transcription, and you need to perform a translation process. That and all you need to do. So for everything you have a function here. For everything you have a function here. And I have taken a data set like a faster code from the internet, means from the database. I have downloaded it. So once it is downloaded, you can see the sequence I'm getting here. I'm converting everything into mRNA. Normally, you cannot do each and everything with the DNA. You need to take a help of your mRNA, means conversion of your DNA to mRNA sequence. Directly, you can do it. We call it as transcription process. You can perform the transcription process and you can get that mRNA. Once you got the mRNA, you can translate. That means you can convert that mRNA into protein in computer. not in real world. I'm talking about only the computer. So how you can do, you can apply the translate function and you can convert that one into a protein. So this is completely worked on your COVID-19 data set. Is anywhere you can find this one. These are the sequence of amino acids what I have in your COVID-19 virus. Yeah. So see, at the end, I try to explore the structure of that protein what we have. I think you can see the protein structure. See, this is what the protein structure of your COVID-19. You can go and you can check what are the amino acids are available and how it is getting folded and all. So this is a very rich area where you can contribute more in your machine learning. If anybody wants, I can share this COVID-19 analysis workbook for you if it is required, or else it is available in internet. So this is a way you can apply your machine learning technique in your bioinformatics. So you want me to explain more about that? I am ready for it, or else you have few questions, I'm ready to answer now. I just want to know the material because I'm not in this major. Yeah, definitely. We'll send you the material. We'll help you with that. The students, I'm open for questions now. Kindly unmute yourself and ask me the questions. I think Rajeshwari raised the hand. Yes.
Okay.
Yes, Rajeshwari.
Hello?
Yeah.
Am I audible?
Yes, you are audible.
Okay, so as we know that protein structure takes an important role for determining the cell activity. Is that mean if AI implement in bioinformatics, that in future will be artificial human maybe?
Rajeshwari, I think that is a bit noisy. Please type the question in the chat box. No? That will be better for me.
Okay. Okay.
Type the question in the chat box.
I have some questions actually, sir. So I will try to untype it.
One second.
And the first one.
Can you type the question in the chat box?
I think it will take much time. Oh God, wait.
It takes much time, huh?
Okay. Structure.
So I just want to know the material because I'm not in this major. I'm ready. Waiting. So I'm.
Comment. Create here. Eyes and so on. So implementation of AI in bioinformatics.
So any other students, they want to ask something?
Is that mean? Okay, that's and second one.
We cannot predict each and.
I can't. Hello? Hello? Sir, I think you are muted. There is. So. Hello? Sir, I think your mic is mute. Where? Kak, kayaknya ke-mute deh.
Okay, Rajeshwari, are you there? Yeah.
Okay.
Look here, each and everything. Look here. Last 10 years, it is like if you want to predict something, predict the structure of a protein, is like up to 60% or 70% accuracy was there. But now we are achieving 98% or 96% nowadays. So the advancement in the technology, we may predict, means we can we can predict something, but I cannot tell you like we can create artificial human. It's a God's creation, right? We cannot
Yeah,
challenge.
I know. Yeah, I know. But if we can learn then predict, then it is not impossible for us to create artificial human actually, sir.
Yes,
Is
yes.
it right?
Look here,
Yeah.
the machine learning, it's look here, what we're doing is completely a block for us. Look here, we are creating a hypothesis, what we have learned, means by using the training examples, we are doing it. But the hypothesis, what machine is creating, it is only understandable for the machines, but we cannot understand it, right?
Okay.
Yeah. We
Okay.
cannot
So,
challenge the
but I
nature.
think,
Nature.
but I think in future it will be. It is possible. It is possible, sir.
Yeah, in future it may happen, but not that much great, not that much accurate. It will happen.
Okay. So, is there any correlation of bioinformatics with biochip, as we saw in movie? Yeah, it is.
You are talking about the movie Biochip, no?
Yeah. Is it any
No,
correlation?
look, biochip is different and bioinformatics is different.
But it took information from
bio is the correlation over there.
Okay.
Yeah, bio is the correlation over there. Look here. So yeah, bioinformatics and biochip is completely different actually. But because in bioinformatics, we are working completely on these biomolecules like DNA, RNA, proteins, and all. So biochips means it is nothing but you are trying to get an inference in between a chip, external computational unit with respect to your brain. So normally now we are doing with that EEG data. We are working with on EEG data, right? So we are taking the data from the human brain. So you know the field of the EEG data we are taking means a lot many research are happening with respect to that area. We are trying to read what is happening inside the human brain.
Yeah.
Yeah. So some inference, some interface is required to understand that. So that we are developing till today.
Okay. Okay. So it is possible. So how do you think it is possible in the improvement of bioinformatics and maybe it will create a biochip, sir? No.
Yeah, of course, definitely.
Okay.
Look here, a simple normally, if a patient is having a cancer or not, how will you identify? How will you detect? Definitely, we'll go with some lab test.
Yeah, lab test. Then now, MRI.
Yeah. Now, what we are doing here, we have a biomarkers for it.
Okay.
And we can take a microscopic image of the cell also, and we can identify whether the person is suffering from leukemia or not. So we are reducing the cost of biological process nowadays. Yes, what is your idea? The machine learning definitely helps your bioinformatics.
Okay.
The biochip.
Okay, so I have last questions. I think I have last question. As you said that in bioinformatics we can predict the structure of like a coronavirus and so on. So can we implement it? Can we implement that in order to predict which immune can invulnerable to virus?
Yes, that is what we call it as. We are using the protein as inhibitors. We call it as inhibitors. Means I have told you, look here, when the
is there any study case for related to that?
Inhibitors. I'm telling you the same thing. Inhibitors.
Okay.
What happens? I'll tell you. Look here, when you have a protein, it is completely structured. Then only we can expect a functionality. So you're going to disturb the structure of a protein. It will not be a functional. Correct? Yes, is it correct? Yeah. Now, what I'm trying to do is when you have a surface protein on any microbes, so you are trying to disturb the structure of the surface protein. Definitely, the functionality it will lose. So why can't you use a protein as inhibitors or as a protection towards all those kind of microbes? Is it possible? Yes.
Is there any study case for that, sir?
Yes.
Is there any study case which related to that?
Yeah. Look, we are doing the research on that kind of area only. Not many advances are happening. Many advances are happening. If I got a next chance to talk to you, definitely I'll tell you what and all we have.
Okay. Okay, thank you for answering, sir. Thank you.
Thank you, Rajeshwari. Novita ma'am, I think any questions we have?
Oh, yeah. I guess no one else is asking. Can I continue the session for closing?
Sir, can
Oh, yeah.
I, sorry,
But
sir, can I get your material for studying purpose?
Yes, I'll share the materials with your organizers, ma.
Okay, thank you, sir.
yeah, I will closing for this event today. Finally, we come to the end of webinar. Visiting lecture. So unfortunately, we already way off the time until 1 p.m. in Indonesian time. So sadly, and we would like to say thanks again for Professor Pavan Kumar and all audience. And I hope we can meet again at other event. Thank you for all audience to join with us until at the last session. Thank you for all audience. Thank you for Professor Pavan Kumar. Yeah, thank you. See you soon.
Thank you, organizers. Thank you, students.
Yeah. Thank you, everyone. Thank you. See you in tomorrow session. Thank you. Bye-bye. Ya, bisa di end.